Welcome to Studious. Today, we are diving deep into one of the most clinically important but often underestimated structures in dental histology, the oral mucosa. The mouth is the primary gateway to the human body, and protecting it is a specialized, continuously maintained lining called the oral mucosa. This tissue is not simply a passive cover, it is an intelligent, architecturally complex, and functionally dynamic membrane. Understanding its architecture means understanding how the oral cavity defends itself against infection, trauma, and chemical assault every single second of every single day. Whether you are preparing for your BDS finals, USMLE Step 1, or dental board examinations, mastering oral mucosal biology will give you a decisive edge. This video will take you through its definition, all 6 of its physiological functions, its 3 major classifications, its microscopic layered architecture, and the 4 variables that determine its clinical color. Stay with us. This is the blueprint your textbooks summarize but never fully explain. Please like, comment, share, and subscribe to Studious.
Let us begin with the foundational definition. The oral mucosa is the moist mucous membrane that lines the entire inner surface of the oral cavity, from the inner lips and cheeks all the way to the soft palate and the floor of the mouth. It is structurally analogous to the skin that covers your external body, but it is specifically adapted to survive in a warm, wet, mechanically active environment. The critical feature that keeps this membrane alive and functional is continuous lubrication. The salivary glands, both major and minor, never stop working. They secrete saliva around the clock, coating the mucosal surface and maintaining a protective, hydrated film. This saliva is not merely water. It contains immunoglobulin A, lysozyme, lactoferrin, histidins, and mucins, all of which collectively form a biochemical shield against microbial invasion. Without this constant salivary flow, the mucosa dries out, cracks, and becomes dramatically more vulnerable to infection, a condition seen clinically in xerostomia and Sjogren's syndrome. So the very first concept to internalize is this: the oral mucosa is a living, lubricated barrier, not a static wall.
Now let us master the 6 distinct physiological functions that the oral mucosa performs to maintain oral homeostasis. First and most critical is protection. The mucosa acts as the primary physical barrier against mechanical trauma from chewing, thermal injury from hot foods, chemical damage from acids and alcohol, and biological invasion by bacteria and viruses. Its stratified epithelium makes this barrier remarkably tough. Second is secretion. Minor salivary glands embedded within the mucosal submucosa continuously secrete mucus and serous fluid, keeping the surface lubricated and the oral environment balanced. Third is sensation. The mucosa is richly innervated and can accurately perceive heat, cold, pain, fine touch, and of course taste through specialized receptor endings and taste buds. Fourth is absorption. The oral mucosa, particularly the sublingual region under the tongue, allows direct absorption of specific drugs and electrolytes into the bloodstream, bypassing the gastrointestinal tract entirely. This is the pharmacological basis of sublingual nitroglycerin in angina and sublingual immunotherapy. Fifth is excretion. The mucosa assists in eliminating certain systemic metabolites outward. Sixth is thermal regulation. Though minor in humans, this function becomes physiologically dominant in canines, who rely heavily on oral panting to dissipate heat. These 6 functions make the oral mucosa one of the most multifunctional tissues in the entire body.
One of the most high-yield topics for your dental histology exams is the functional classification of oral mucosa into 3 types. And the key principle to remember is this: structure always follows function. The first and most widespread type is the lining mucosa, also called reflectory mucosa, which covers approximately 60% of the oral cavity surface area. It is non-keratinized, making it soft, flexible, highly elastic, and capable of stretching and relaxing without damage. This flexibility is essential because lining mucosa must accommodate the constant movement required for speech, mastication, and facial expression, without participating in the grinding of food itself. The second type is the masticatory mucosa, which constitutes 25% of mucosal surface area. It is keratinized, packed with tough, opaque keratin proteins, making it firm, highly resilient, and resistant to the high compressive forces generated during chewing. The third type is the specialized mucosa, covering 15% of mucosal surface. Its structure is mixed, containing both keratinized and non-keratinized regions, and its defining feature is the presence of distinct papillae and taste buds. It is dedicated exclusively to sensory function, specifically the perception of taste. A useful mnemonic to remember the proportions: 60, 25, 15. Lining, masticatory, specialized.
Now that you know the 3 mucosal types, let us map each one precisely to its anatomical location in the oral cavity, because exam questions will absolutely test your ability to match classification with region. The lining zone, representing the majority of the oral surface, covers the labial mucosa of the inner lips, the buccal mucosa of the inner cheeks, the floor of the mouth, the ventral, that is the underside, surface of the tongue, the soft palate, and the alveolar mucosa that drapes over the bone beyond the gingival margin. All of these regions need to move freely and flexibly, which is why they are non-keratinized. The masticatory zone is restricted to 2 high-impact anatomical sites: the gingiva, meaning the gums, and the hard palate. Both of these regions are pressed firmly against teeth or bone during every single bite of food, so they demand the structural toughness of keratinization. The specialized zone is located exclusively on the dorsal, that is the top surface of the tongue, where the mucosal surface is studded with papillae of different types. The filiform papillae provide texture and grip. The fungiform papillae are vascular and red, and the circumvallate and foliate papillae house the taste buds that detect sweet, salty, sour, bitter, and umami. This anatomical functional mapping is a cornerstone of dental board examinations.
Let us now go microscopic and examine the layered histological architecture of the oral mucosa. The oral mucosa is built upon 3 tissue strata, stacked one upon another. Layer 1 is the epithelium, the outermost shield and the most immediately visible component under the microscope. The oral epithelium is classified as stratified squamous epithelium, and this is an absolute exam essential. The word stratified tells you it is built from multiple stacked layers called strata. The word squamous tells you the surface cells are flat. The deepest layer is the stratum basale, where mitotically active cuboidal cells continuously divide to replenish the surface. Above that is the stratum spinosum, characterized by spiny, desmosomally connected cells that provide cohesion and structural integrity. In keratinized regions, the stratum granulosum and stratum corneum are added on top, with cornified cells packed with keratin filaments. In non-keratinized regions, the stratum corneum is absent, and surface cells retain their nuclei. Layer 2 is the lamina propria, the core connective tissue layer anchoring the epithelium from below. It is rich in collagen and elastic fibers, blood vessels, lymphatics, and nerve endings. Layer 3 is the submucosa, a variable, optional layer present only beneath flexible mucosal regions. It contains adipose cells, glandular tissue, and muscle fibers that allow the mucosa to move independently of the underlying bone or cartilage. Our final and perhaps most clinically fascinating topic is the color of the oral mucosa. Because color is one of the most powerful diagnostic tools available at chairside examination, and understanding why the mucosa appears the way it does requires knowledge of 4 intersecting physiological variables. Variable 1 is melanin concentration, the amount of natural pigmentation produced by melanocytes in the basal epithelial layer. Higher melanin concentrations produce darker mucosal tones, which is a completely normal physiological finding in individuals of African, Asian, and Mediterranean descent. Variable 2 is the degree of keratinization. Thicker, more opaque layers of keratin act as a physical screen that masks the underlying red vascular bed, making the tissue appear paler and more white-pink, as seen in the gingiva and hard palate. Variable 3 is blood vessel state, the proximity of lamina propria vessels to the surface and their degree of dilation. Dilated vessels bring more hemoglobin close to the surface, intensifying redness, which is why inflamed mucosa turns bright red, a sign of acute gingivitis or mucosal pathology. Variable 4 is epithelial thickness. A thin epithelium allows the deep red color of the underlying vascular bed to dominate, explaining why the ventral tongue and sublingual mucosa appear intensely red, while the thickly keratinized hard palate looks pale pink. Clinically, any deviation from the expected color, pallor, erythema, leukoplakia, or hyperpigmentation is a red flag demanding histological investigation. That is your complete oral mucosa blueprint. Please like, comment, share, and subscribe to Studious.